Flannery from the Novartis Institute of Tropical Diseases in California, and she's going to be talking about using the humanized mouse model to study Vivax relapse.
All right, well, thank you, Jessica and Kevin, very much for organizing this. Jessica did really do a lot of good work to make sure that we could talk very concisely about a topic, relapse, which we don't get to dive into that often. So I'm going to be presenting work on behalf of the team that was done in the last 3 years at the Center for Infectious Disease Work in Stefan Keppe's lab with Sebastian Mikolajczyk. Is this the, how do I get this?
The green one, yeah.
Okay. So the ways that we've always looked at relapse in the past is we've used the human model and the monkey model. And so if you think about what we're actually looking at, we're looking at patency in the blood. We're looking at parasites in the blood. We're not going in and we're not looking in anyone's liver to see whether they actually have hypnozoites or not. And I think this is really important because, as you'll see with a lot of the work that we've done, this has been a real challenge because we haven't necessarily known really what to expect if we were to go in and look inside of these livers. So with these two models, we have learned a tremendous amount in the past century, as Kevin pointed out. And just to remind you, we're always following relapse in clinical trials and in monkeys. So we're infecting the patient or the monkey, and then we're looking for that patency at a later point in time. So you can see that the primary infection is usually after about 8 to 10 days, and then at some point later there's a relapse that's observed, and this can vary, the periodicity can vary based on geographic region. And in these trials, what we're doing is we're actually then treating with a drug, and when we're treating with a drug, we're not treating prophylactically. We have no way to know if there's parasites in a person's liver. We have no diagnostic for hypnozoites. So the only way that these trials are actually completed is to go, and if someone actually has a patent infection in their blood, then they get treated with the drug primaquine to clear out their hypnozoites, is the theory, right? So this means that the parasite, the hypnozoite, has been there for at least 10 to 11 days because there's already a patent blood stage infection. So because of this, we don't know a lot about the parasite itself and what's actually going on in the liver. And so what we wanted to do, and the group started at CIDR by Sebastian and Ashley, was to actually use the liver chimeric humanized mouse to be able to look at the parasites actually in the liver. And there are a lot of advantages to this model. So the way these mice work is they're an immunocompromised animal that's a triple gene knockout. They actually need to be supplemented with a substrate so that the mouse hepatocytes can survive. And when you withdraw that, the mouse hepatocytes will die, and then you can add primary human hepatocytes, inject them into the spleen, and they will repopulate the animal's liver. And so we can see up to 90% of the liver becomes humanized. So these are all human primary cells. And so in this model, we're able to actually go in and look at the parasites. So obviously with the monkey model, you can take the livers out, but it's much more challenging to section and look through the liver for all these parasites. We give the mice a very high inoculum of sporozoites, anywhere from 500,000 to 1 million. We can do this by mosquito bite or by tail vein injection. And this allows us to get a very robust infection so we can actually go in and look at these parasites. So what I'm showing you here are hypnozoites and schizons, both at an earlier time point, day 7. So this is during the primary infection. So you see a hypnozoite and a schizon, and then we also can observe hypnozoites and schizons at day 21. How do we know it's a hypnozoite versus a schizon? We base this on size, UIS4 staining. We always see this prominence with UIS4, the parasitophorus vacuole membrane protein marker, and by having a single nuclei. So that's how we decide that it's actually a hypnozoite. And Sebastian has done a great job with developing, and colleagues at CIDR, developing a lot of antibodies, so we actually can look at many different organelles in the parasite and try to get an idea of what's actually going on with these hypnozoites, as Kevin said. So what we can also do is we can actually measure the size of these parasites. So what you can see here is you can see that these are the schizonts, the primary infection. They grow exponentially. And what we can see is by day 3, we can see that differentiation between a hypnozoite and a schizont. We can't tell as early as day 2, but by day 3, we have prominence on all hypnozoites, and we see that size discrepancy. This graph here, only the days that we've looked for this specific experiment are labeled on the graph, because every time we go in and look, we actually have to sacrifice the animal. And we've gone out to 49 days, so we haven't tried any longer, but we've done a 49-day infection, and we looked in the liver, and we were able to find hypnozoites. So another thing that we see at any of these later time points is we see schizonts, and we don't see nearly as many as we do during the primary infection, but they're definitely schizonts, and so the conclusion you would draw is that these are actually the relapsing schizonts. So to actually show this, we used one of the same tools that Sandra discussed. So we actually did an infection. Again, this is on mice, and this is with all isolates from Thailand, again, collaborating with Jutseman's great group. And we used the same PI4K inhibitor that Sandra mentioned, and so we know that this molecule, if it's given day 5, 6, 7, and 8, to the animals, we know that it will actually kill all the schizonts, but it won't kill the hypnozoites. So we can essentially purify for a hypnozoite-only culture. And so when we do this, and then we look at day 8, so we can look at the primary infection, and then we can look at a later time point, and you can see here where we have no schizonts in the 048 treated group, then we do see schizonts in the later time point. So these are relapsing hypnozoites. There are no schizonts on day 8, but if we look at a later time point, they're definitely relapsing. We can also look in the blood, so we can just look for RNA, and we can look for merozoites egressing. We could repopulate these animals with reticulocytes and see the liver to blood stage transition. It's a lot of work, so we actually just use qRT-PCR with Sean Murphy at UW, and we're able to very robustly tell whether there's just merozoites egressing or not. Now, this is just a snapshot. It's only that time during the day that we're taking the sample, but you can see that we don't get anything in the blood here during the primary infection with the 048 treated, and then we definitely then get the relapse. We see it in the blood at the later time points. So a few more of the more interesting things that we've been able to see and show is you can see here we kind of had this anecdotal evidence early on. When we work with Jutseman for historical reasons, the parasites are always genotyped for their CSP alleles. So in Plasmodium vivax, there's 2 CSP alleles. And they're genotyped, and we know whether they're VK210 or VK247. We've done this study now with 10 VK210 or 9 VK210 isolates, 9 VK247 isolates. And we actually see a statistically significant difference in the number of hypnozoites that form. So this is the hypnozoite formation frequency, so percentage of liver stages that are hypnozoites. You see that we get a higher formation rate in VK247 isolates. This is likely not because of CSP, potentially a gene that's linked with CSP, but we don't know what's causing this observation. Another thing that's very interesting, we've wondered for a long time, we've worked with the Badia lab and the Kyle lab for a couple of years now, and we always see that in vitro there seems to be more hypnozoites, and we've always wondered if this was based on the strains and the geography or if it was platform-based. And we've now been able to we've been able to use the same isolate where we infected the mice and the in vitro cultures with the same isolate. And on both occasions, we consistently see that the hypnozoite formation frequency is higher in vitro than it is in vivo, and that's consistent. We also see that the hypnozoite size in vitro is actually larger than the hypnozoite size in vivo, but then that is reversed for the schizonts, so it's quite interesting, and we could talk maybe in the end about what we think is actually going on there. The other really important biological thing that this model has showed, this is really important, so this actually recapitulates some data that was done, some experiments that were done in the 50s by infecting humans with Plasmodium vivax, and What you can see here is that we're just talking very short primaquine treatment. So obviously primaquine treatment in the clinic is 14 days, but if we just do this for 1 or 2 days, we see this really interesting phenotype where you can see if we start treating it at day 0 or 1, we don't see any schizonts or any hypnozoites in the animal's liver. So primaquine will prevent the formation of hypnozoites. It's a prophylactic. But if we then take this dosing out a little further, and then once we get to day 3 and 4, if we give 2 doses on day 3 and 4, we don't see any difference in the number of hypnozoites in the 3-4 treated compared to the controls. And this is really important because this suggests that something at that time point, there's some sort of commitment to whether it's a mature hypnozoite, there's something going on that makes that hypnozoite no longer as susceptible to that short course of treatment, as well as potentially this would have the same effect with other prophylactic drugs. So that's a lot of the biology that we've done with the model. We also are using the model to test for potential novel radical cure candidates. So this is how we're currently using the model in that way. So what we actually do is we run it out to day 24 because we want to make sure that we are having a latent infection. We also want to treat exactly how they're treating in the clinic with primaquine and chloroquine. So we use them in combination because we know that there's some suggestion that they're synergistic, and we could argue about that for hours, but we just want to treat exactly how they're doing in the clinic so that that's not any question that we have. And then we also treat with the PI4K inhibitor to clear out all those schizonts, because we know if we bring primaquine on 4 days after infection that it will actually arrest those schizonts and they'll stay there, so that will confound our results. So that's the reason that we use all 3 drugs as our positive control. And you can see here that this is looking at liver 18S RNA copies of the parasite in the liver, and this obviously doesn't differentiate between schizonts and hypnozoites, but what you can see is that both primaquine, chloroquine, and the PI4K inhibitor, we get a reduction in the number of liver stages that are there at day 24, and with the triple therapy, we actually see complete reduction in parasites that are in the liver, which is not something we necessarily expected. The number of hypnozoites, we don't see any infection. We've repeated this twice now with the triple therapy. We see a decrease in the number of hypnozoites with the primaquine chloroquine and the 048, the PI4K inhibitor, and then schizonts, we don't see any with the 048, and we have this observation of the primaquine chloroquine, where in one case we don't see any schizonts. With the other isolate we did, but we went and looked at these morphologically. They don't look like parasites that are alive. They look like they're stalled and probably dead, but they're still there in the liver. So the last thing that we've used this model for is also to think about biomarkers. So as I said, that we have no way to know if someone out there has a hypnozoite in their liver, and this is going to be really important when it comes to eradication. Because if we can't find a diagnostic, we're going to have to mass drug treat large populations to try and get rid of these hypnozoites. So using the mice, we've gone in, this is done in collaboration with Hernando Del Portillo, who's an exosome expert, and we have used the mouse serum and looked in the mouse serum to look for parasite proteins with the idea that those exosomes are being shed from the liver and that they could serve as a diagnostic for latent hypnozoite infection. So this is very preliminary data, but proof of concept that we can detect HSP70 and histone 2A in all samples in the serum. We can also see this protein that's in Plasmodium vivax, not annotated as LISP1, but potentially LISP1, which is a liver stage protein. So this is suggesting that we can absolutely identify these proteins. They're present in the infected mice. They're not present in the uninfected mice. It's also a great platform to actually look at human proteins that maybe are changed as well, and they're only liver-derived human proteins because that's the only human cells that are in the mouse. So in that same study, we were able to show that in infected mice, 19 of the 23 human proteins that were differentially expressed were actually confirmed in that same P-Vivax study that was done in humans, where they looked at proteomes of infected versus uninfected individuals. And then we're also using the mouse to try and find liver-specific exosome markers, which don't currently exist, but this would obviously allow us to really increase the sensitivity of these biomarker assays if we could actually go and look for liver-specific exosomes. So I'll just summarize that this animal model can really be used to go in and look and see what the parasite is actually doing in the liver. It recapitulates exactly what's happening in the monkey and the human. If we only want to look at relapse, we can run those studies in the same way. I think the day 3 hypnozoite development is really important. I think for a while we're really confused about we could easily prevent hypnozoite formation. That's not that. We have a lot of drugs that can do that, but it's really these latent hypnozoites which we're trying to get rid of. Stress may actually induce the hypnozoite formation. That's a hypothesis based on using that exact same strain where we see differences in vivo and in vitro, and also coming off of some of the transcriptomes that have been published as far as what's expressed in the hypnozoite. So that's an area worth further study. And then it's obviously a great model to look for biomarkers and drugs. So I'd like to thank everyone that contributed to that. It's a large number of people to be able to logistically do this. And I would also just like to mention that Sebastian and I are now at the Novartis Institute for Tropical Diseases. We're continuing to work on liver stages and look for radical cure drugs. So if you want to know anything about that, you can talk to me afterwards. Thank you.
There we go. Yeah. Questions for Dr. Flannery? Great.
Nice talk.
Thanks.
So I'm curious about the difference you saw in hypnozoite conversion rate between VK210 and 247 alleles. You suggested you think it's actually a gene other than CSP that mediates that phenotype. Do you want to?
Yeah, I mean, so we don't. I mean, based on what we know about CSP, we can't really hypothesize how CSP is affecting the formation of hypnozoites. I mean, we could definitely come up with things, and it's possible. It's more likely, potentially, that it's actually just linked. It's also quite interesting that we just essentially randomly picked a marker, and we see an association. That's quite intriguing, but I totally agree. It could actually absolutely be CSP itself. There's no reason that it couldn't be.
Nice talk, covering everything in basic science, really to the clinic. I think what you're saying is that prophylactic primaquine only has to be there for the first 3 days, because after that, the hypnozoites are made, and that's what Alving et al. Showed in the 50s. What you could do was now use tefenequine for exactly the same purpose, and it would have immediate clinical benefit, because it would tell you how long after the last time of exposure of a person to Vivax tefenequine has to be given in order to prevent hypnozoite formation. That would be an immediate translation from your work to the clinic and to the field.
Yeah, thank you. Yeah, we absolutely think that although a major focus is to go out there and treat people that have hypnozoites that are already formed in their liver, there is also immediate need to prevent those hypnozoites from forming. So we want to make sure that we're actually using prophylactic drugs to prevent hypnozoite formation, because that's going to knock down a large number of what transmission is out there. Recent paper showed that 90% of blood stage infections are actually due to relapse, and that's causing transmission. So, yeah.
Erika, I should know this, but I have to ask.
I don't believe you.
I just forgot. When you use atovaquone in the prophylactic mode,
yeah.
Do you prevent the formation of hypnozoites?
Do we prevent formation? No, not in our model. And actually, it's interesting that what you said, Kevin, about atovaquone is I left the atovaquone data out because it's very confusing, because it's not what we expect to see, of course. We do see that we don't get schizons forming, but we see actually an increase in the number of hypnozoites. So we think that maybe there's some sort of stress response that's actually causing these hypnozoites, and they do look like real hypnozoites.
Hi, Erika. In the exosome data, which I think was recently published, and
Yep.
you've shown here today, the detection of LISP1 was something that you mentioned
Yes.
was detected in all of the infections. Do you see LISP1 in any small forms? Is it possible is there an antibody to LISP1? Is it lighting up?
Yeah, so we tried that. There was an antibody that was produced for the Western work that was done in that paper. We did not see any reactivity in our schizonts or our hypnozoids, and so we don't know if that means that it's actually not there, or if, you know, the antibody just doesn't work in the sectioned hepatocytes. But we are working further on it, because if it was something we could detect, the more proteins we know that are in the hypnozoite, the better.
And number 2, LISP2?
LISP2 is expressed in the hypnozoids, so it's expressed in a subset of the hypnozoids. And the idea, so this is a lot of work that was done by a postdoc at NITD, Devendra Gupta, and he showed that a subset of the hypnozoids express LISP2. And the idea, the hypothesis is that those are actually activated hypnozoids, because that's, as you know, a late liver stage protein. So that may actually tell us, even though it still looks like a hypnozoite, that hypnozoite is ready to start going through schizogony.
And that's inside the parasite or outside of the parasite?
It's both. It's inside and seems to be membrane associated. Yeah.
Okay, great talk. Regarding primaquine treatment, so you give about 30 milligram per kilogram even for the few days that you give,
Yeah.
right? When you do this treatment in vitro for all those in vitro models, you're using reasonably high concentration of primaquine as required for getting rid of the hypnozoites.
Yeah.
That doesn't seem to be the case when you're doing this in mice. That the actual concentration that you reach is not going to be that high. Am I missing something, or is that the equivalent to what you see in Sangeeta's or Dennis's in vitro models, where you require very high concentration of primaquine, versus what happens in vivo in your humanized liver model? Yeah.
So the 30 mg per kg is not extremely high, but is on the higher side for treating mice, especially for 14 days. But we've measured the levels in the blood, and the whole problem, right, is we don't know what the active metabolite is. So we see that primaquine goes away, and you keep pulsing it with the primaquine. I do think, so in vitro, I think there's more things going on in vivo, and potentially it's the host and all of these other, redox reactions, all this that's going on that's actually facilitating primaquine to work better in vivo. But I will also say that we never see complete clearance of all these hypnozoites, and I think that's a product of just giving so many sporozoites, and I think that's true in vitro as well. If you think about the number of parasites that are actually being introduced into the liver and becoming hypnozoites, it's on the magnitude of 5, probably. And so if you have a certain amount of efficacy and you think of how long it takes those parasites to relapse, then if you take those numbers out and you look at the 200 that we have in our models, it might be that you only actually do get a reduction in the number of hypnozoites.
And the implications for this drug screening in vitro versus in vivo could be significant in this case.
Yes.
Awesome. Thank you.